Hello dear students, welcome back once again to Kamala Online Learning. We are back again with another informative video lesson from your schedule. Please be completely attentive during the teaching. Keep your textbook and also a rough book with you. Note down important points and difficult or new words as we go. This video lesson will begin with an overview which will give you an idea about the contents of the lesson. It will be followed by learning objectives. Please note down the learning objectives in your notes and try to answer them at the end of the session. If you have any doubts, rewatch the video and refer to your textbook and study material. In case you need further clarification, please mail your doubts to kamalaonlinelearning@gmail.com. This video is recorded with simple, easy to understand language and eye-catching graphics. We hope it helps you to understand the concepts better. So, let's get started.
Welcome to the chapter story of Microorganisms Part 1.
This slide presents the overview of the chapter. Learning objectives. By the end of this chapter, you will be able to
Understand the different types of microorganisms. Explain the microscopic invention. Know the discovery of microorganisms. Know the different types of microorganisms found in pond water. Understand the organisms present in bread mould. Know the bacteria present in buttermilk. Explain the protozoa present in hay decoction. Know the different types of microorganisms present in soil and their importance. Explain the diseases caused by bacteria. Understand bacterial staining. Know about viruses.
Introduction. Before entering into the chapter, follow the instructions shown on the screen.
Microorganisms are small living creatures that cannot be seen through our naked eye. There are 4 types of microorganisms, which include bacteria, virus, fungi, and protozoa. Microorganisms are also living organisms which feed, grow, and reproduce themselves. Do you know? Why do we add some buttermilk drops to lukewarm milk to make curd? Why does the food get spoiled after a few days, even after cooking? Why do we get a bad smell from our mouth after we wake up in the morning? Let us try to find out the answers for these questions and learn more about the different organisms that are involved in causing such changes. Let us study about the invention of microscope and discovery of microorganisms. Most of the people wondered why the changes occur in food and what will be the reason for getting bad smell from our mouth early in the morning. Antony van Leeuwenhoek is a scientist who was born on October 24th in the year 1632. He is the father of microbiology and also the first microbiologist. He built a powerful microscope with a single lens which could magnify the object 300 times. His curiosity and skill of making powerful lenses made him invent a powerful microscope. He discovered small moving organisms in 1678 with the help of his microscope. He named them as animalcules and later changed it to bacteria. Later, he also observed many microorganisms under his microscope and gave their respective names to those organisms, which helped him to discover many other microorganisms. Let us learn about microorganisms. A few microorganisms can be seen and some others cannot be seen with our naked eye. The unseen organisms are called microorganisms, which can be observed under the microscope. The images on the screen show different microorganisms. Here, let us study about groups of microorganisms. Microorganisms belong to some of the groups like bacteria, fungi, protozoa, algae, and certain arthropods. Let us study about these microorganisms with the help of some activities by using a microscope.
Knowledge check. Attempt the following questions to check your understanding.
Let us observe the microscopic organisms present in the pond water by performing an activity. Collect the samples of pond water along with the greenish scrapings. Add 1 or 2 drops of pond water on the slide and cover it with a cover slip. Place the slide under the microscope and observe it. Dark green colored structures are observed under the microscope. From the above activity, we have learned that different types of microorganisms are present in pond water. They include Spirogyra, Chlamydomonas, Spirulina, etc. Now, let us know about fungi. You might have observed some small umbrella-like growth between the grasses in a field, over rotten materials of dumped waste, and on the edges of wet, rotten wooden planks during rainy season. You might have also observed white patches on the bark of the trees where we can find some fungi. Let us observe the fungi on bread mould by doing an activity. To observe fungi on a bread mould. To perform this experiment, the following materials are used. They are spoiled bread, permanent slide, plucker, cover slip, and microscope. Take a slice of spoiled bread and a permanent slide. Now, place some rotten part of the spoiled bread on a permanent slide by using a plucker. Next, add a few drops of water on the slide and cover it with a cover slip. Whitish thread-like growth with masses of black, grey and green fine dotted structures are observed. The tiny thread-colored structures that are found on the bread are generally called mould. This mould develops on the spoiled material and it has the capability of producing new mould. The common bread mould plant consists of fine thread-like projections called hyphae and thin stems having knob-like structures called sporangia. Sporangium in singular. Each sporangium contains hundreds of minute pores. When the sporangium bursts, the tiny spores are dispersed into the air.
Knowledge check. Attempt the following questions to check your understanding.
Used to perform this experiment. They are glass of buttermilk, dropper, crystal violet, permanent slide, cover slip, lamp and microscope. Take a glass of buttermilk and a permanent slide. Now, add 2 to 3 drops of buttermilk on a slide with the help of a dropper. Heat the slide slightly on a lamp for about 3 to 4 seconds. Next, add a few drops of crystal violet on the slide and cover it with a cover slip. Leave the slide aside for 30 to 60 seconds. Next, wash the slide with water. Dark blue-colored structures are observed from the microscope. The small, dark blue-colored structures are the lactobacillus bacteria present in buttermilk. Now, let us study about algae. We can find greenish pond water in our surroundings. The growth of algae and other plants in the water changes the color of water to green. Chara, Spirogyra, etc. are some of the algae which can be seen with our naked eye. But most of the algae found in water are microscopic and can be observed only through a microscope. To observe the algae present in water, let us perform a small activity. Let us perform an activity to observe algae in pond water. Collect some water from a pond which appears dark green in color and contains filamentous structures. Put 1 or 2 filaments on a slide. Next, cover the slide with a cover slip and place it under the microscope. Observe the slide under the compound microscope. Green-colored string-like structures are observed. The green-colored structures observed are called spirogyra. About Protozoa. Protozoa is the other group of microorganisms which are found mainly in water and soil. To observe Protozoa more clearly, let us do an activity. Collect some pond water in a bucket and hay, dried grass, legumes or other herbaceous plants which are used as animal fodder. Now, soak the hay in pond water for 3 to 4 days until it becomes hay decoction. On the 4th day, place 3 to 4 drops of hay decoction on a permanent slide. Next, cover the slide with a cover slip and place it under the microscope. Different types of organisms are observed. Different types of organisms like Paramecium, Spirulina, Serratium, Cyclops, Daphnia, and some other kinds of bacteria are present in the hay decoction. Here, let us learn about microarthropods. Some of the microarthropods are very important for the soil as they help in increasing the soil fertility. They increase soil fertility by decomposing the biomass through digestion, which converts the bigger compounds into smaller compounds. These arthropods are also found on our skin, eyelids, beddings, rugs, etc. Diseases like scabies mites are caused by arthropods. Arthropods are not microorganisms like bacteria, but they are minute-sized arthropods as they are joint-legged organisms. Let us study about the types of microorganisms present in soil. Soil is rich in microorganisms which contain thousands of microorganisms like bacteria, fungi, protozoa, microarthropods. Almost 5.5 tons of fungi and bacteria can be found in the top 8 inches of soil of 1 acre area. These microorganisms are very helpful for the growth of crops. Excess usage of pesticides on crops kills these bacteria. We can observe these bacteria through microscope or by growing them in different media. Collect some soil in a beaker. Add some water to the beaker and stir it well with the help of a spoon. Wait for some time until the soil particles settle down. Next, add 2 to 3 drops of water on a permanent slide and cover it with a cover slip. Place the slide under the microscope and observe it. Different types of organisms are observed. Thus, from the activity, it is clear that the microorganisms are also present in the soil. Thus, we can say that there is another wonderful world present in living organisms. Let us study other places where we can find microorganisms. Diversity of microorganisms. Microorganisms are present everywhere around the world. They are found in water, air, and in the soil. They are also present inside the bodies of human beings, animals, and in plants. Microorganisms can survive in all types of environment, ranging from ice-cold climatic conditions to hot springs, deserts to marshy lands. Some microorganisms grow on other organisms like parasites, and some may exist independently. Let us know the diseases caused by microorganisms. Some of the diseases like typhoid, tuberculosis, and septicemia, blood poisoning, are caused by bacteria. Some of the skin diseases are caused by fungi, bacteria, and arthropods. A few diseases like malaria, amoebiosis are caused by protozoans. Here, let us study about viruses. Viruses are very different and interesting microorganisms. When they are outside of a living cell, they behave as non-living organisms. But they behave as living organisms when they are present inside the living cell. They reproduce in the same way as bacteria and other plants and animals. These viruses are visible only through very powerful electron microscope. Polio, swine flu, conjunctivitis, smallpox, chickenpox, and AIDS are some diseases which are caused by the viruses. Bacterial staining. Bacteria are very small and tiny microorganisms. Before seeing bacteria under the microscope, we must stain them. Spread the bacteria on a slide and slightly heat the slide. Next, add a few drops of crystal violet to the slide. Wash the slide gently after 30 to 60 seconds and dry it. Now, see the slide under the microscope in 25x or 40x.
Keywords. List of keywords are shown on the screen. Summary. Let us recap the highlights of this chapter.
Microorganisms are present everywhere around the world. They cannot be seen through our naked eye. We can only see microorganisms through microscope. Antony van Leeuwenhoek is a scientist born on October 24th in the year 1632. He is the father of microbiology and also the first microbiologist. The tiny thread-colored structures found on the bread are generally called mould. Hyde and Schultz in 1997 discovered two types of bacteria, and one of the biggest bacteria, Margarita namibiensis, is found in coastal waters of Namibia. Some of the microarthropods are very important for the soil as they help in increasing the soil fertility. The viruses are visible only through very powerful electronic microscope. They lie between living and non-living organisms. Viruses can reproduce only when they are inside the cell.
Test your understanding of the lesson by taking the mock unit test.
You have successfully completed the chapter Story of Microorganisms Part 1.